All right, hi everyone. My name is Aisley Johnson, and today I'm going to be talking to you guys about the evolution of the cell. And so this includes the theories of the mechanisms that first drove evolution. Okay, as an introduction, I'm going to talk about in this presentation how the cell evolved. And so there's many theories surrounding how the cell evolved. The most popular and well-supported theory is endosymbiosis, but there's also other theories out there, including the inside-out theory, the hydrogen hypothesis, as well as the theory of micromutations. Okay, so I'm also going to be talking about who contributed to these theories, and I'll mostly be talking about who contributed to the theory of endosymbiosis because this was the most important one. And so this includes Konstantin Marechkowski, Ivan Wallin, and Lynn Margulis. Finally, I'll be talking about why cell evolution is important, and just as a sneak peek, some of the things I'll be talking about are its application in scientific fields, just knowing how the cell functions and how it helps in those scientific fields, as well as looking into future evolution and how things might evolve in the future, looking at the mechanisms of, how evolution first worked. Okay, so the cell that exists today is much different from the first living cell. And so the first cell is hypothesized to kind of look like the one on the right that you see. And this is just super simple. It has a strand of RNA molecule as well as a membrane around it. And this model is actually really similar to the prokaryotic cell. Okay, and then on the left we have the modern prokaryotic cell and the modern eukaryotic cell. And so prokaryotes are bacteria and eukaryotes are protists, fungi, animal, and plants. And within those animal cells and plant cells are going to be much different from each other, but they share the same basic features. And then I'm just going to talk to you a little bit about the differences between prokaryotes and eukaryotes. So you might be able to see some of these on the screen, but the prokaryotes are actually much smaller. They're around 5 micrometers, while eukaryotes are around 10 micrometers. Prokaryotes contain their own circular DNA, and they replicate by binary fission, while eukaryotes have DNA arranged into histones and replicate by mitosis or meiosis, which you guys have already probably learned. And then another important difference is that eukaryotes contain membrane-bound organelles. And so some of the most important organelles of the cell are the chloroplast and mitochondria. And so the chloroplast only exists in plants, so that's important to note. But both of these organelles are the powerhouse of the cells. And so they both conduct cellular respiration. So chloroplasts conduct photosynthesis and mitochondria conduct oxidative phosphorylation. And that's just, they're different methods of providing energy to the cell. And that makes them unique to other organelles as well. Another unique property is that they both contain their own DNA, and it's circular DNA. Next, they both have double membranes, and then they're both aerobic, which means they need oxygen to make the energy for the cell. Okay, let's talk about the contributions of these different scientists. So the first one is Konstantin Marechkowski, and he was a botanist that proposed the idea of symbiogenesis. So this theory states that complex organisms were the result of beneficial relationships between more simple organisms. And so Ivan Wallin, he took this theory and he applied it to the cell. And so he said mitochondria were simple organisms. They began as bacteria, and then they became part of the cell because of this theory. And then finally, Lynn Margulis, he expanded on these ideas even more and said that mitochondria were once free bacterium, and they became engulfed by larger cells, and the cells benefited from the mitochondria because they provided energy, while the mitochondria benefited from the larger pre-eukaryotic cell because it offered protection. And so these were some of the contributions they made, and this helped us develop the theory that we have now, the theory of endosymbiosis, which is defined as the relationship between two organisms where one lives inside the other because it's beneficial for both organisms. So you can see in this picture, in the first event, we have a proeukaryotic cell, and then we have aerobic bacterium. That aerobic bacterium is the mitochondria. And then in the second event, we also have a photosynthetic bacterium, which is the chloroplast. And so these become engulfed into the cell. And then you can see on the right the modern day eukaryotic cells, both plant and animal. Okay, so let's talk more about the role of the mitochondria and chloroplast in the evolution of the cell and why they provide evidence that the cell evolved this way. So we talked a little bit about the advantages. Both offered cellular respiration, energy to the cell, and then the cell provided protection to both of these organelles. But the evidence begins with the method of replication. So before this, I should note that scientists thought that if they could prove that both these organelles were similar to prokaryotes, then they could prove that they were once on their own and then they were enveloped. And so each piece of evidence shows the similarity between prokaryotes and these organelles. So the first is going to be the method of replication. All of these organelles and the prokaryotes, they replicate by binary fission, and then they all contain circular DNA. Okay, and then the next piece of evidence will be the size. And so they all range from 1 to 10 microns in diameter. And then lastly, they all have a double membrane. And so you can see in this picture, the organelles had their own red membrane, and then when they were enveloped into the cell, they gained another membrane, which is the blue one that you see in that picture. And so this provides most of the evidence for the endosymbiotic theory, but of course there are alternate theories regarding the evolution of the cell. And so classically, as we discussed, scientists believed that the eukaryotic cell had a nucleus and then enveloped the mitochondria and chloroplasts. Others believe, however, that the prokaryotic cell enveloped the mitochondria and chloroplasts and then acquired other eukaryotic features. So within these two options lies a lot of different orders in which the cell acquired the other eukaryotic features, but those are more variants of the endosymbiotic theory. But these three are much different. So the first one is the inside-out theory. This theory proposed that mitochondria lived on the outside of the cell, and then the cell developed kind of these protrusions that kind of reached around it, and they fused and enveloped the cell that way. And then the gaps between the protrusions became organelles such as the endoplasmic reticulum. And so this is different because it says they acquired the organelles by protrusions, while the endosymbiotic theory proposes engulfment. Okay, the next one is hydrogen hypothesis, and this basically flip-flops the endosymbiotic theory. So this states that they actually had aerobic hosts, so the cell was aerobic, and then that the mitochondria were anaerobic, meaning they did not need oxygen to produce energy. And so this theory kind of gets its evidence by explaining the common ancestry between mitochondria and hydrogenosomes. And hydrogenosomes are actually just anaerobic mitochondria. So it just kind of explains the common ancestry between these two much more than the endosymbiotic theory. And then it also says mitochondria lacking eukaryotes never existed. So they just think that the eukaryotes always had mitochondria because it was so essential to the development of early eukaryotic cells. Okay, so the last theory is going to be the theory of micromutations. This probably has the least evidence, and this theory proposes that there was numerous micromutations that accumulated over time because they were advantageous to the cell. However, this one isn't well supported because we know that a lot of the times evolution often works horizontally instead of vertically in a progressive manner. And so, but, discussing these theories also helps us understand other options of how the cell evolved. But again, endosymbiosis is the most well-supported theory. And so, if it's so advantageous, I ask, why did it only occur once? And so the mitochondria and chloroplasts overcame the issues, but the reason why it only occurred once is because the more complex a cell becomes, the more energy it needs. And in addition to that, engulfment takes up a lot of space. And so it's super rare because of the high cost that it comes at. But of course, mitochondria and chloroplasts overcame these because they provide their own energy for the cell. So it's more of an advantage than a disadvantage. And so the evolution of the first simple cell included the engulfment of the mitochondria and chloroplasts and grew in complexity in the future, but prokaryotes remain the simplest life form. Okay, so in conclusion, I'm just going to talk a little bit about the significance that cell evolution has and why it's important that we discuss the evolution of the cell. So first of all, discussing the evolution of the cell helps us search for these mechanisms of how the cell evolved and all other kinds of life. So scientists can look for symbiosis as a mechanism of evolution, and they can also search for the role of metabolism that plays in evolution and how improved metabolism can be fixated so rapidly because that's what happened with the cell. And then in addition, a deeper understanding of how the cell functions is really going to help us in all other fields, because understanding how it replicates, how it achieves energy, these all can be applied to medical field, biochemical field, ecologically. You know, we've already studied genetics and the role of that in environments, and obviously in the medical field we need to know a lot about the cell to be able to help people. And then lastly, it's always important to look to the past and kind of see how things evolved in the past to help us in the present with these current things, but also prepare for the future and see how cells might evolve in the future if they do ever more. And that is the end of my presentation, and I hope you guys enjoyed it. Here are my references. And thank you guys so much for watching.